I wanted to show everybody an alternative tool to the APE software. I know APE has a little bit of a learning curve and it sometimes is not intuitive. Although, you could get the job done in APE, I preferentially prefer, if that's a thing, to use this tool here called Benchling. Now, Benchling is an online lab notebook, and many lab spaces in academia and also in industry use this lab notebook to keep track of all their work. And it also has a slew of cool molecular tools and things you can use if you're doing genomic work. So I'm going to go over with you guys an alternative. So I'm going to be posting an announcement, and on the announcement, you're going to have an invite link to the group. Now, one drawback from Benchling is that you have to sign up for it. It's completely free. But I know some people just don't want to be able, don't want to sign up for things and things like that. So if that's you, that's okay too. You can just keep using APE. Anything Benchling can do, it can also be done in APE. So let's get started. So once you accept the invite link and sign up, you're going to have a folder called Genetics and Genomics Shared Project. You want to come up in here. You're going to see the PTC project. You want to get in there. And then here, genotyping, let's go in there. Just follow the yellow brick road. I recently just made this folder called test, but you're going to make your folder maybe with your name on it. So how do you do that? You just click the little plus here and click folder. And the folder is going to create. It's going to ask you to name that folder. And voila, you're going to have a folder. Now go into your folder. We're going to be using the test folder here. And then what you're going to do is, this is your space. So I'm going to go over also, there's going to be a 2-for-1 special here. I'm going to go over the stuff for the TAS2R38 gene in reference to building primers, getting a genomic sequence onto Benchling and how we go about that. In class 5, the pre-videos for class 5, you saw how it was done for APE. So we're going to do a little bit the same thing here for Benchling. So again, click the little plus sign and you're going to go to DNA RNA, and we're going to want to import DNA RNA sequences, right? So you're gonna click import, and then you could either have a file like we did with APE. We get the gene bank file, and we can open it that way, or we can import it directly from database, right? And you could also upload APE files here, all these different types of file types. So that's cool. You could either select chromosome regions and the species you're looking for, or we can import it from a database, which I like. So we know our gene of interest, right, is TAS2R38. Right, let's see if it finds it. So we're going to do that sequence. We're going to click enter. Okay, and then it's going to say, what species we're interested in. Human. All right, we're going to click search. It's going to look for it, and hopefully, boom, voila, it found it, and it has all the details, all the specifics about it. What's also really cool about Benchling is that you can actually specify if you want any upstream or downstream bases flanking the genomic sequence or your gene itself. So it's really cool if you say you want to do a CRISPR experiment or something very genomic-y that you need to play around with the DNA around the gene. This tool is perfect to do that. But let's carry on. We're going to import this thing. Load up. And then boom, it imports to your folder. Again, make sure you're importing it to the correct folder. And look, it's all annotated. It has the protein, the translated proteins just depicted in different colors. It has the exons denoted, and it has the whole genomic sequence also annotated. So it's pretty cool. So question 1 on the pre-class homework last time, the last one, right, was what was your expected base pairs for the PCR product, right? So I saw a lot of people struggling with that question, and probably we're a little bit turned around on how to go about solving it. Um, we're gonna do it in Benchling. So let's see. What you would need is you would need your primers, right? Because we're gonna do a PCR product, and this is important too because you're gonna be building your own primers very soon. So take this as also another example. So what we can do is we can actually go to our lab manual, right? And so we have our module manual, and here are the primers you need, right? So we're going to do the reverse one first because the forward primer, as those of you may know, had something special, right? So we're going to copy this sequence here. Let's see if this works. Copy. Okay. And then we're going to go back to Benchling, right? And then we're going to click up here for the find buttons, and we're going to paste our bases. And we're going to not allow for mismatches for right now. So, okay, so 0 mismatches. Here's our primer sequence, right? So let's click this. We could actually click this. We can close this, and then we can actually create an annotation. I see that you could create a primer here. I don't like this feature because it creates another element, but you can play around and see what you like. So I like to go to annotate, and then I'm gonna say, hey, this is, remember we're doing the reverse primer first, so it's reverse primer, all right? And then we get to pick a color. Let's make this, keep it brown. And the forward, remember this is the reverse primer, so it's gonna go in the reverse direction in this case. And we're gonna save that thing. And voila, there you have it. Your reverse primer is on your sequence. So that's cool. So now let's get the forward in there. So same thing, we're gonna go back to our manual. We're gonna copy this sequence here, copy, and then we're gonna go back to Benchling, Control Find or little magnifying glass. And sometimes if you're clicked in like this and you try to find, it's gonna say 0. So you wanna make sure nothing's highlighted and paste your sequence. Uh-oh, says 0. Right? And those of you that figured it out for the homework, great job. But the primer actually has some mismatches in it. So in order to get this thing to bind, is you have to allow mismatches. So if you allow one mismatch, there it is. You have your primer binding sequence there. So same thing, we can click this sequence, close out of here, create, and then let's do a primer. Oh, well, an annotation. And then I'll keep it the same color as the forward, as the reverse, but this time we're going forward. And we're gonna call this a primer. Okay, so voila, save that thing. And now you got your forward and reverse primers. Now watch this, right? So we're gonna close this. Boom, we click the forward primer and we drag all the way to the reverse primer, right? And this is our PCR, right? This is what we amplified off this gene. You see its length, 303, right? 303. Now,